video, and I need you to go check out some other resources for the steps of meiosis. I'm not actually going to go through the steps of meiosis at all. So here's where you want to go. First of all and foremost, I think the most informative thing you can do is go to the figures on page 244 and 245 in your book and spend some serious time writing down what you see happening in those and what it says are happening in those diagrams, and there's definitions in there, and that's really important. If you want a step-by-step seeing it happen in animation, then go to our website, to the resources for Unit 4, and there's an animation section. These two animations are great. The animation, Steps of Meiosis animation, gets into about the detail that our book does, and then the unique features of meiosis just gives, it's a real brief deal with a little bit of extra information about meiosis. So pause your video now, go check those out. Okay, now you should have a really good grasp of what's going on in meiosis and how the cells go from haploid to diploid, or diploid to haploid, sorry, and just a decent idea of what's going on. I just remember last year from questions that students had, sorry, when we went through this in advanced biology, is the terms that are on the screen right now were really confusing, and it seemed like a lot of terms for the same things and whatever. So I'm just going to kind of buzz through these really quickly and try to show you what they mean and when they happen and some things like that. So first of all, homologs, we've talked about that a bunch of times, but if we look here at homologs, those are the when we were looking at the karyotype, these are chromosomes that are the same size, same shape, they stain the same way if we put stains on them, and they code for the same types of genes. So if you're looking at these here, you'll notice that I have like A, B, C down the side, or the picture has A, B, C down the side of this one, and A, B, C. That just means gene A, gene B, gene C. They're in the same locations on these chromosomes. When you're looking at diagrams in the book and they're using different colors, a blue color and a red color, what they're showing here is the blue-colored ones are always the chromosomes that came from this person's dad, or the paternal chromosomes, and then the chromosomes that came from this person's mom are the maternal chromosome. And then if you want to think through that at the end of meiosis, if you go back through and look at the pictures, at the end of meiosis you're giving a combination of your mom and dad's chromosomes down to your offspring. So grandma and grandpa's chromosomes are going down through to the offspring. But anyway, so these are homologs. When we talk about synapsis, so in prophase, when chromosomes start, and I'm not sure how this is going to work for me, but when chromosomes start to appear in prophase 1 of meiosis 1, the chromosomes, they're first of all, they're not just spread around everywhere on the cell. The homologs are together, and they're not just together, but they're actually like overlapping, so you can't really distinguish the two chromosomes from each other. That's synapsis, okay? And there is a, what's the term, synaptonemal, synaptonemal, anyway, synaptonemal complex, a bunch of proteins that are lining both chromosomes all the way up and down, and these chromosomes, there's a video that I used to watch in general biology that said that the chromosomes embrace each other. They kind of wind around with it. It was very touching. They were like, and the chromosomes embrace. Anyway, during this time of synapsis, and this is where people start to get confused, during the time of synapsis is when crossing over can happen. Okay, so I'm going to bring another picture up here to kind of show if this synapsis, and I'm just going to throw it over top of everything, this picture, let's say we zoomed in on those two chromosomes. During synapsis, okay, there's going to be places where chromosomes are going to be crisscrossing a little bit, okay, and during synapsis what can happen is these things at the exact same spots, they can actually trade sections of chromosome, and they'll actually break at the exact same spot. So if it's these two, it'll break here and it'll break here, breaking at the exact same spots, and they'll exchange the ends of their chromosomes. No genes should be lost here. They're going to be breaking at the exact same spots. Then we'll keep the same types of traits being passed on. But this can happen several places. So it could also happen up here. Anyway, it can happen several places on the chromosome. Not predictable at all. So this is a huge source of genetic, mixing genetic information up a little bit. But that's crossing over is when they actually swap information. So I'm going to delete this picture. So let me just say that one more time. It's not just the fact that they're crisscrossing that makes it crossing over. Crossing over is when they actually swap genetic information. So if I get rid of these two chromosomes, take me a second here. Okay, here's two chromosomes that they have, and if I pull them apart, they have actually swapped information, so crossing over has happened. Okay, and you can see that part of the red chromosome is now on the blue, and part of the blue chromosome is now on the red. Crossing over has happened. Okay, so that's crossing over. That happens during synapsis. Okay, so if I take, let me straighten this one back up. When we were in synapsis here, these two were just pretty much embracing, and during that time crossing over happens where they're swapping information, and then at some point towards the end of prophase, as we're moving into metaphase, the synaptinimal, anyway, those complexes, those proteins are going to start to break apart, and these will then start to separate to the point where we can actually see the two chromosomes. That is what we call a tetrad. So as we're moving towards metaphase of meiosis 1, right here, a tetrad will form. The reason it's called a tetrad, the prefix tetra, means 4, and we've got 1, 2, 3, 4 chromosomes, chromatids, that are lined up together. Oftentimes, when you look at a cell that is in its tetrad form, they will still be close enough that the places where crossing over happened, let me see if I can do this, the places where crossing over happened, they'll still be overlapping, and they were crisscrossing, if you will, but they'll be overlapping, and that is the chiasma. Okay, and so this cell, or this set of chromosomes, these homologs, they have one chiasma happening here, where they are still, this is where crossing over had happened, the swapping of the DNA had happened, but they are still crisscrossing each other. That's called a chiasma, or chiasmata, if there's several of them. The plural is chiasmata. Okay, so then we get into metaphase, and these things are lined up in the, these things are lined up on the metaphase plate. Sorry, I keep bringing my hand in the way in order to move these things. Anyway, in the metaphase place, so they're lined up there, and the significance of this, of the crossing over and whatever, is that if we continue to move forward with what we look at on meiosis, we will find that this chromatid here and this one right here, they are no longer identical. Like when I explained the homologs at the beginning, homologs were identical. This chromatid, sister chromatids were identical. They aren't anymore because we just swapped some genetic information. So what we've done is we've added another form of variation that we could produce. The production here is either eggs or sperm. We could produce a sperm that has this set of information, and in this case it's a red A and a red B and a red C. Or we could produce a sperm with a red A and a red B and then a blue C. Or we get a blue A, blue B, red C. Or you're seeing the difference here. So we're adding variation to what can go in the next, into the next set of, the gametes that are made. If you push this out through 46 chromosomes or 23 pairs of chromosomes, chiasmata, crossing over happening at various times on different chromosomes, differently every time, it really, really amplifies the amount of genetic variation you can get through meiosis. So hopefully that helps with some of the difficult parts. If you're stuck on like the stages of meiosis, go back and look at the diagram in the book, go back and watch those animations, or ask me some questions. So that'll be it for this, and I hope it was helpful, and you have a great evening.